Hello everyone, I welcome you all here again. Today we have very special guest with us, Dr. Mantosh Kumar, who is joining me from NIH in USA. So NIH means the National Institute of Health. He is doing his postdoc there. So before welcoming him, I want to just introduce him, give a background about him. He is basically from Bihar, which is a state in India, and he did his master's in Kerala, and after that he started his PhD in Rajiv Gandhi Center for Biotechnology in Trivandrum, Kerala, and his PhD was in about the calcium signaling and the neurological disease, and after neurodegenerative disease actually. Sorry for that. And then he joined his postdoc in National Institute of Health in USA. So we welcome him today, and we will try to understand so many things from him. But first thing will be about the career advice, how he reached to the National Institute of Health, which is the kind of top institute in USA, and we will also talk to him about the science, because he's working in really interesting topic, which is neurodegenerative disease and calcium signaling. So I'm very much curious to know about these things. So welcome, Mantos, and let's start our talk.
Thank you, Dhiren. It is quite a pleasure to talk to you and talk to your audience, actually. So, as far as what is or how is my journey, as you have already mentioned it, I started my neuroscience interest in second year of my MTech course, that is master's course in India, and then carrying forward it till my PhD in RGCB, Thiruvananthapuram, India, where I basically worked on calcium signaling and how it affects neurodegenerative diseases, mainly actually Alzheimer's disease,
Mhm, mhm.
and or in very specific instance, it is called like cytotoxicity induced disorders. So then I wanted to move ahead in the same direction. So I approached some labs, and I selected a lab in National Institute of Health, which is working on Parkinson's disease, which is a uh, 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 center, which is a research center which is focused on Parkinson's disease. And here, basically, what I am looking at is how some particular type of neurons are getting actually generated, and how they are connected to other neurons locally and to different parts of the brain, and how it affects the behavioral aspects, motor deficits and motor functions in the experimental animal. Actually, here I'm working on mice, so I'm looking all
Thank you so much for telling me about these things. And before we go in the details of your research topic, I want to be in like a journal career thing. So because your journey is really interesting. I'm also from UP and you are from Bihar. So when I see myself, I feel like it was long journey, right? We learned so many things. We fought with so many struggles, and then we reached here. So it's a kind of quite a big experience for us. So I especially want to go in the detail of the your experience, research experience, and how your journey was. So my first question is, when you started your master's, so how difficult was it to coming from Bihar? So you were from Bihar and then going for masters and then going for PhD and then to reaching to NIH. So how was this journey?
Yeah, sure. I can. Yeah, sure. Definitely, I would like to tell you about my story. Actually, if I tell you frankly, in my village, that is a small village in Patna, Rama. So in the village, there were hardly any PhD. So I don't know up till now whether I was the
So
first
in your
one to
village
register
there
for
is
PhD.
no one who
No,
studied like
yeah, no one till
highest.
my knowledge actually. So coming from that background where you do not have that much knowledge about higher studies and how you go ahead with your PhD and all. So as I remember, even my relatives used to tell me what you are doing, and I'm telling, okay, I'm doing science. I mean, I'm doing research. What is this research? So it was like, but definitely they were my parents were more curious, more focused for my study. They were knowing whatever I will be doing will be good for the family and maybe good for other aspects in the society. So it was a little bit struggle, but as people are there to support also, so I got support, and I was. I mean, people suffered my endeavor throughout these so many years. Actually, it is, I mean, took long time. You do masters, and then people start looking for jobs. So it was, I mean, after masters, you are supposed to get a job, you earn something, but you are registered for PhD, especially in India. You get join a PhD, and it is not a very financially you will not be strong. So it is you have that aspect also to look for. What in India it takes almost 5 to 6 minimum, that much amount of time. So then as I finished my masters, then but one thing was sure that I was interested in neuroscience from my
I mean,
at
you
least
were
from
you
my
were totally
masters.
focused on your way that you want to reach
Yes,
there. So that was
yes,
from
yeah,
beginning.
that was there.
Okay. Yeah.
Yes, that was
Beautiful.
there, and I had chances to join other labs also,
Hmm.
which were. I mean, some people say it was good, bad, whatever. But I had chances for joining other labs also. But I chose some particular lab only because of my interest in neuroscience. So it was. I mean, interest was there, and of course there you have to I mean you have to act persistently and you have to work hard. That should also be there. If you falter in between in the journey, then your destination is not going to be there. I mean, you may not reach there. So it was of course that aspect also. Then if you are asking how was my feeling throughout, good, bad, both. So it was some aspect when you, I mean, in masters, I mean, in PhD, you do not, I mean, you start PhD, you look for, oh, I will discover something very big. But at the end of PhD, you will come down to the fact, okay, this is the what you will get after 5 years of work. So, but if you are focused in your area, then you will definitely keep on looking. What next I have to do? So I was looking for the next journey. I was looking after this studying calcium signaling in general neurodegenerative disease. What I look for. So I was either looking for areas which are maybe Alzheimer's disease or multiple sclerosis, stroke, or Parkinson's disease. So I finally decided to go ahead with Parkinson's disease. So I mean, it is a good, bad, sweet, bitter journey, but this is how life goes.
Yeah, really nice. And I'm really happy that you are working in a reputed institute now, because coming from this background and struggling, and then struggle pays off when you reach the place you wanted to be. Right.
Yeah, thank you. Yeah.
Yeah, so I'm also curious to know when you started applying, when you were in like last year of your PhD, that time you started making your application, and why did you choose NIH?
Yeah. So yeah, I mean, it is a very valid question and very important question for anyone who want to pursue career in research. So after, I mean, just after submitting my thesis, PhD thesis, I immediately started applying to my labs which are relevant to my field. So I started many labs. I started applying many labs, almost around 20, 25 labs I approached. Some labs replied positively, and out of that, some offered me a postdoctoral fellowship. Out of that, I mainly chose because NIH because it was relevant to my interest, and I wanted to pursue my research career in this direction. So then I chose, okay, we'll go ahead with NIH, and I just went ahead with that.
So when you are making your application, what was the important thing which you are taking care of, like mostly taking care of in your CV, in your motivation letter?
Yeah. So one thing is that you have to highlight your positive aspects of your research experiences in your throughout your whatever you have achieved so far in research, any specialized training course, any presentation award, any travel award, any international travel award, any national funding. So all these you have to highlight in your CV. And regarding motivation letter, you have to let the other person know about your previous background experience in research. So, what are the techniques you used? How you addressed your PhD question, and how you want to go ahead with all this set of skills which you had in your research career till PhD. How you are going to use that in your next level research means postdoctoral research. So, in the motivation letter, you particularly have to link your research skills so far achieved and use it in your future research projects. Again, you have to be very particular about writing to different labs. You have to address each lab separately and highlight their trajectory throughout their lab history, what they started working on, and then till now what they are achieving, and how I am going to fit into that story. So this is very important. You do not have to put a general application to every lab. You have to be selective, and you have to be very careful when you are writing motivation letter to different labs. You should be different, really different. It should
Yeah.
not be the same everywhere.
Yeah, so I agree. I agree with all the points you said. It's really important to be very precise, very accurate, and very point to point when you're making your CV a motivation letter. And also important to read about the lab and to know about the
Yes,
lab
yes,
before when
yes,
you're
of
applying
course.
it. True. So let's from career we are moving now to the research. So it's really interesting. I like your research because neurons. Everyone like the function of brain, the neurons, how they are connected, how the signaling is going on, because these things are really, really important for our body function, and also they are really interesting same time, right? So I want to know about your research a bit. What do you do in your PhD, and what you are doing in an IH now, and how relevant it is in general life?
Sure. I'll start with my PhD. So my PhD research was focused on calcium signaling and how it is relevant for preventing neurodegeneration. So we call it neuroprotection. So how calcium signaling is relevant for neuroprotection. So basically, what happens? There is the neuronal disorders. Most often, there is calcium dyshomeostasis. So that dysregulation affects many aspects of the cell, especially in this case, neuron. And then they let the neuron die. So I wanted to prevent it from getting dead. So we worked on a particular kind of calcium channel called NMDR receptor, and we wanted to prevent excessive activity of NMDR. So we used many synthetic and naturally derived compounds to do that. And actually, NMDR function, normal function is necessary even for memory, learning, and everything. But excessive activity of particular type of NMDR. Actually, I worked on gluon 2B gluon 2B containing NMDR, which is more relevant for excitotoxicity. So then, how it can affect the the therapy for these neuronal disorders. So I just went ahead with the same theme. Now leaving the excitotoxicity aspect, now I move ahead for my postdoctoral research in the field of Parkinson's disease. That is also a kind of neurodegenerative disease. Actually, it is the second most abundant neurodegenerative disease. First
Mhm.
one being the Alzheimer's disease. So in this lab, which I am currently working in, they are focused on actually Parkinson disease. There is selective degeneration of a particular subset of neurons, which is basically substantia nigra pars compacta dopaminergic neurons. So they are getting degenerated specifically. And as degeneration starts there, then progresses to other areas also. So it is a progressive disorder. And we want to see.
Sorry, how it progresses. You mean that this, if one neuron is damaged and then it infects other, how the, I
Yeah,
want to understand.
yeah. Actually, there is a protein dysregulation. As in case of Parkinson, it is alpha-synuclein. So they get become oligomer, then become fibril. Some oligomers are can pass through the neuronal membrane. They go to other neurons. Then this progression happens, and it will be like a series of events. Starts from one place and then can go ahead for disrupting all throughout the down the line. So it will start degenerating in substantia nigra pars compacta, but as the disease progresses in the late stage of Parkinson's disease, you can see degeneration in other areas of brain also. But it started with substantia nigra pars compacta. So what I in this current lab we are looking is how these selective neurons which got degenerated, that dopaminergic neurons in substantia nigra pars compacta, they got degenerated, how they are connected to other areas of the brain, how they are getting projections from other neurons, or how they are projecting to other neurons. Then we want to see how it affects this disruption in projections, or their connection or network connectivity, how it is affecting motor functions in the brain. So we in our lab we use mice for studying all this. We use many imaging techniques and behavioral experiments to address all these questions. So this is how in general I can tell you
So
that
so
my
what
current
I
lab is
understood
working.
is that you are trying to understand the connections between the neurons, and through these connections you want to track the disease, right? So if
Yes, see, tracking is yes one aspect, but we are not working in diagnosis or of course for clinical setup it is important, but we are looking if this particular neuron is dying, what is happening? Neuron is not an isolated unit. Neuron is connected to hundreds of other neurons, even thousands of other neurons through
wow, it is a
synapses.
kind of circuit.
Yeah, it is kind of circuit, and if one unit of the circuit is disrupted, then it may affect the whole circuit in some way or other way. So we are particularly focused on how since one unit of the circuit is disrupted, how it is going to affect the other unit, whether it only affect motor function, whether it affect cognitive function, whether it can affect any other physiological function in the brain. So we are, of course, we are mapping how the connectivity. It is kind of connectome. So how the connectivity throughout the brain is affected if the selected neurons, or selected dopaminergic neurons are degenerating in Parkinson's disease. So that is if that is affected, then how it is affecting motor function. Why we are studying motor function? Because Parkinson's disease is mainly known for motor disorder, movement disorder, problem in walking, problem in locomotion, problem in rotation, problem in hand and leg movement, tremor. Actually, this name was called cerebral palsy. It's kind of shaking stroke. Stroke is there, but shaking. Patients of Parkinson's disease have uncontrolled, non-voluntary. It is like they are not willing to move hands, but still their hands are moved.
Yeah, let me ask you one question. How prevalent this problem is in the whole world, and specifically in India? Do you think the India is also having so many big cases of this disease, neurodegenerative diseases?
Yes, it is related to aging. Older population, wherever there is a larger proportion of older generation people, there this disease is more prevalent. In USA, many more older people are there. In India also older population is increasing. So, I mean, we expect, although diagnosis plays a factor, if a diagnosis is not happening, we may not assign some person or some this person is Parkinson's disease affected person. But if diagnosis is done properly, we will discover definitely many patients with Parkinson's in India also. So the common theme is like that. If older population is more, chances of getting Parkinson's or Alzheimer's even, all these neurodegenerative conditions are more prevalent in older population. So wherever there is older population, in USA it is quite old population.
Can I ask
In
you?
India it is young.
Yeah, in India.
In India, our population is relatively very young.
Yeah,
More
yeah.
than 50% population are young. I mean, more than, I mean, between 30, below 35 or something. So, but in here, older people are more in the population. So here, prevalence is also more.
Is there any genetic link also with this disease, and is just age related? I mean, if you are getting
Yeah.
older, probably you have more
So,
probability of disease. Are there is any genetic link, some other links also with this disease?
yeah, there is genetic link, both in case of Alzheimer also and Parkinson's also. There are many genes which have been discovered that it is but that is not a single gene disease. It is called polygenic disease. So if one genetic mutation or genetic disruption happens, we cannot for sure tell, okay, this person is highly prone. We can say, but it will develop Parkinson. Maybe we may not so. We may not say so. It is for particularly for Parkinson's, more disease are called sporadic disease, not genetic. It is it is called environmental induced factors,
Yeah, yeah.
which can be pollution, which can be the food we eat, which can be the way the social strata we are in. If we are poor, it may affect more. If we are having junk food, it may affect in a relatively different manner. So at least for Parkinson's, the genetic aspect is not that very established. It is there. Some risk genes are there, which we can say in summary, it is a polygenic disorder. It may not be because of single gene, and then disease is progressing. So it is the genetic link is there, and that is more important when we are pre-screening patients. If we are finding, oh, this particular gene is there, then we will think, we have to follow up this patient. I mean, this person for maybe 5 years, 10 years down the road, how they are getting symptoms or not. If we diagnose this early, it is very good, because most of the symptoms, especially motor function disorders or motor deficits, happens in the late stage of this disease. That is, it is very difficult to manage clinically.
So is there any medicine which is available? What is the drug development thing which is going on in this field? So how near
Yeah.
you are to treat the disease with the drugs, with any other therapy?
Unfortunately, we do not have a cure for either for Alzheimer or Parkinson's. So, I mean, it is an active area of research. People are, I mean, there are some symptomatic drugs. For Alzheimer, there are some drugs which are, which have been useful so far. Likewise, for Parkinson's also, since I was telling about dopamine neuron are getting lost. So what we want to do, replace the dopamine. So we externally give the dopamine called levodopa. So that is one aspect, but that won't cure the disease. It is just a symptomatic strategy. Then there are people who use deep brain stimulation. They put electrode in the brain, they stimulate particular area of the brain, and then sometimes that is also helpful. Then there are some stem cell therapy. If dopaminergic neurons are getting lost, so people take a skin cell from the patient, make it pluripotent. I mean, then put in the brain, like it will work as a dopaminergic neuron.
Wow.
So these are the areas where things are going on, but still very not established way. I mean, we cannot say well, this is the regimen you give and the patient will get cured.
Wow.
No. So still in the developing field.
Mm-hmm. And I have one more question which I wanted to ask you is about the techniques which you are using in your research. So, which kind of techniques you are using? Suppose you want to see the activity of neurons. As you say, the neurons having a kind of circuit, right? So they are really interconnected with each other. And if you have in a specific area in the small part of the brain, you have kind of thousands of neurons which are giving signals from one to each other. And
Sure,
let's say our
sure,
body
sure.
is one part is connected with the other part, right? Through the neurons. So how do you understand this language? Because it's really complicated, right? So
Yeah, sure.
what do
Yeah,
you
this
do in
is
the lab so to understand these type of signaling and these type of language?
yeah. Structurally, we can see by imaging, like how they are connected to each other. That we can see by imaging, but functionally, that we can see by recording. That is
Mhm.
electrophysiology. So in this lab, we are establishing electrophysiology unit to record the functioning, like to see how they are functioning. If they are connected to this set of neurons, how they are functioning here, whether they are spiking more, whether they are spiking less, whether there is dysregulation in the spiking pattern. So these all functional aspect we can see by recording. Biochemically also we can see what things are going upregulated, downregulated, that we can see. But for structural integrity and structural, this if there is a disruption, then we can use viral mediated neuroenvironmental tracing. We inject a virus with a probe, which will infect the neurons, and then that will wherever the neurons circuit are elongated, that much we can trace. That is one aspect. We can do that it in live mice also. That we use called fiber a technique called fiber photometry, where we in the live animal we can implant a optic fiber and see the fluorescence coming out of that
So you
area.
put the
So
sorry, you put the optic fibers in the brain of mice and then you record the activity.
yes.
Wow,
Yeah,
nice.
so for before that, we what we do is we put first we put the virus, let it infect, let it spread the reporter protein, for example, green fluorescent protein, and after maybe 2 3 weeks, we put fiber, we drill another hole, we put the fiber, and then we get the signal of fluorescent signal. If it has gone to that point, then signal will come. If it has not gone to that point, then signal will not come. So we assume that okay, this is not connected to that point if there is no signal. If that is connected to that point, means there will be fluorescent signal. So by that way, we like kind of make a trajectory from here to here that has gone, then it will give signal in photometry, fiber photometry. So it is happening in the live brain. Sometimes it happens in the behaving animal also. If the animal is walking, how it is having, if it is having, active also in that condition also, we put, with the fiber is there, and we can see the fluorescence up and down happening as a wave. So
Mhm.
we know which extent they have activity there also.
Wow, really nice. So you record the brain activity like in a kind of live, in a kind of live.
Yeah, yeah, yeah.
You can see the graphs that
Yeah, we can see.
that
Yeah, in
in
the
the
behaving mice, we can see. Yes.
how neurons are connected and how they are really nice. I really like this type of research where you can just see these things which are going through in the neurons which are really small, right? So
Yeah.
coming out of the research a bit, how you feel because now you are working in IH since, if I'm right, if I'm not right, please correct me because
August 2020.
yeah, like yeah, from 6 months, right? You joined in August.
Yeah, almost 6 months.
So
Yeah.
how do you feel in IH? I know this Corona time is not the same as the research used to be before, but how do you feel coming out of India and these 6 months? What was the how good NIH is?
Yeah, actually, as far as my research center is concerned, in India where I did my PhD, infrastructure wise, there is a lot of good infrastructure in my research center RGCB. I am using the same stereotaxy setup here. I'm using the same flow cytometry. I have been trained in IIT Chennai in the same electrophysiology setup, which I will be establishing here. So in terms of infrastructure, I do not find very much difference. Of course, there is funding problem. Here, funding is not a problem. But as far as other research aspects related activity, we see here little bit more openness and I mean, here also people are working individually, but in terms of research output, they feel the urge to get consultation from other lab members or other PIs in the department. So, which I think may be improved in back in India. So there, people are very. I mean, like if my paper is coming out, I may not be willing to share with all the members in my research institute. But I mean, here I see openness is better.
Really nice. So, uh, I think that we will stop our talk now, and it was really nice to discuss with you about the career, about your journey from a state of Bihar, which is considered a kind of like we UP and Bihar, my state also, we are considered as a kind of not in the middle also in India. Like we are still progressing. We are not too progressed state in India. We are doing
Yeah.
our work. So from there to reaching in the top institute in USA is it is amazing. I know how much hard work you have to put it to reach there, right? So Dr. Mantos, I'm really thankful that you gave me your time and you discussed so many things about your research, about your career, and it's going to be really helpful for the people who are watching us, who probably, and then yeah, thank you so much
Yeah,
again.
thank you, Dhirendra. It was a very good experience for me also, and I very much enjoyed talking to you and then talking to your audience. And I hope your audience find it useful and they can get something good out of it. And I wish best of luck to everyone who is watching and to you also. Thank you
Thank
very
you so
much.
much.